Welcome everyone. I'm Mike Fave, a future nurse practitioner and independent health researcher. I'm here to guide you through deep dives into complex research and physiology to bring you key answers for your health. Today we're diving into the story of an explosive duo silently wreaking havoc on our bodies. This duo is the combination of omega-6 polyunsaturated fatty acids, the highly reactive fats hiding in our everyday foods, and reactive oxygen species, the unsuspecting spark triggered by many aspects of our everyday lives. Together, they form a dangerous combination that may be leading to the explosion of cardiovascular disease we are seeing in the modern world. But don't worry, we're here to disarm this ticking time bomb and provide you with the key things you can do to minimize your exposure to this destructive combination. So, without further ado, let's jump in. So here's the basic summary of what we're going to be covering today. And don't worry if you don't know any of these terms yet. We're going to define them as we go. I'll explain what they are as we go. And what I want to point out before we jump in is that this presentation is building on the previous presentation that I did covering the actual mechanisms of plaque formation in cardiovascular disease. So that presentation will give you a nice context to further understand what I'm discussing in this specific presentation. So first piece here is increased reactive oxygen species stress, or increased reactive oxygen species, plus increased omega-6 and PUFA here is polyunsaturated fatty acids in LDL. So this is LDL cholesterol, the cholesterol that people normally think is the bad cholesterol, increases oxidized LDL, so essentially damaged LDL. So what a lot of people don't know is that LDL doesn't necessarily cause cardiovascular disease in and of itself. LDL has to be oxidized first to cause cardiovascular disease. So what we're seeing here is the combination of these two factors leading to increased formation of oxidized LDL. And then with the oxidized LDL, what it does is it actually damages the vasculature and increases immune activation. And the chronic exposure to these processes leads to the formation of atherosclerosis, or plaques inside the arteries. So let's talk about the oxidized LDL hypothesis of heart disease. So there's a little blurb here. I'm going to read it, and then I'll describe what that blurb is with this graphic. So the low-density lipoprotein, or LDL, oxidation hypothesis gained traction during the 1980s because it was noted that in general, native unoxidized LDL cholesterol does not cause foam cell formation. In other words, LDL had to become oxidized first in order for atherosclerosis to develop. So essentially, regular LDL, the normal cholesterol floating around in your bloodstream, doesn't cause atherosclerosis, or it doesn't cause immune cells to take up the fats and form something called foam cells. And if you want to understand how that actually occurs, I did describe this process in my previous video, so I'll link that video inside the description. What happens is the LDL has to get oxidized first, or essentially damaged, and then once it's damaged, then the immune cells are able to recognize it, pick it up, and basically they're trying to clean out these damaged or problematic LDL cholesterol. So essentially what's going on here, I'm going to zoom in a bit. You have normal LDL particles. So this, as you can see here, native lipid molecules. So this is just fats. And you have basically fats they don't mix well with water. You can kind of think of oil and water. So in order to transport fats around our bloodstream, we actually needed to create, or the body needed to create, some type of ingenious system. And what essentially they use lipoproteins. So you can see ApoB is a lipoprotein. And the lipoprotein interacts with the different lipids and forms this spherical structure, this what we would normally call our LDL cholesterol, and it allows these fats to be transported around the bloodstream. Now, these fats, depending on what their composition is, and that's what we're going to get into, can become damaged. So you can see here the red ones are oxidized phosphatidylcholine or phospholipids, and then the other different colors in here are other oxidized lipid products. So these fats can get damaged by multiple different factors inside the body. We'll talk about what some of those are in specific relation to reactive oxygen species. And then essentially, when these components get damaged, they can also damage the protein. So this apoprotein or apolipoprotein B, the little blue lines around here, can get damaged from the oxidized fats. And then when it gets modified and the fats are also oxidized, you get an oxidized LDL particle. Now, what they're saying here is that the oxidized LDL particle can be recognized by these very specific receptors. And these receptors are basically recognizing this damaged cholesterol molecule. And that'll be important as we go along. So this is a general or a very general overview of what's going on with LDL. And we're going to break this apart into much more specific pieces. So as we get here, now what we're going to talk about is the direct toxic effects of LDL on the vasculature. So what the researchers say is to say increase local and systemic levels of oxidized LDL, induce endothelial cell activation, dysfunction, apoptosis, and impaired vasorelaxation, and contribute causally to atherosclerosis development and progression. In endothelial cells, oxidized LDL signals mainly via the lectin-like oxidized low-density lipoprotein receptor. Yeah, you can say that 10 times fast. If they abbreviate it here, just LOX1. And activates diverse cellular second messengers, including the inflammatory pathway, nuclear factor kappa beta, and AP1, two oxidative stress responsive transcription factors important in regulations of cytokines, chemokines, and adhesion molecules. Some of the induced cytokines also activate nuclear factor kappa beta and AP1 and can reinforce the inflammatory signaling cascade. So essentially, what you're seeing here is oxidized LDL interacts with the endothelial cells. You can see the endothelial cell, its membrane here. You can see oxidized LDL. And when it interacts with this endothelial cell, it interacts with this receptor, LOX1, and essentially it causes a whole bunch of negative effects. So some of these you can see are this death, programmed cell death. So essentially the endothelial cell commits suicide. Foam cell formation. So the immune cells come in and they gobble up this oxidized LDL. Macrophages are those immune cells. So that's where you see the foam cell formation. And then also endothelial activation. Some of the other things they talked about is it induces inflammatory cascades, and the inflammatory cascades can reinduce themselves. So it becomes this feed-forward process. So oxidized LDL, and something that they discuss here specifically, is causally related to atherosclerosis. So it's not an associative factor. These are mechanisms where it is directly causing atherosclerosis. So it's a very important piece in the picture. So we want to understand what is forming this oxidized LDL. But first I wanted to show you that it is indeed causally involved in atherosclerosis. And then we're going to go here to the next slide. So just furthering this process with oxidized LDL, the oxidized LDL activates immune cells. So what I want to walk you guys through here and describe is how oxidized LDL is interacting with the immune cells to form atherosclerosis. So the researchers start off here. They say the other important result of our work is that oxidized LDL can co-activate at least two types of tissue-resident cells, macrophages and mast cells. So both of these are immune cells. This co-activation induced macrophages and mast cells to release two powerful pro-inflammatory mediators, tumor necrosis factor alpha and histamine. So essentially, oxidized LDL interacts with these immune cells, and then it triggers the immune cells to release these very pro-inflammatory components, tumor necrosis factor alpha and histamine. If anybody is familiar with some of the other videos that I've done, or some of the other topics that Jay and I have talked about in the Energy Balance podcast, tumor necrosis factor alpha is also released in response to endotoxin, which inside the bioenergetic sphere is often understood to be one of the main toxins to watch out for, and that usually is derived from gut issues. But you're also seeing this as a function of oxidized LDL signaling some of the same cascades or inflammatory pathways. The next thing they say is as a pro-inflammatory mediator, tumor necrosis factor alpha triggers nuclear factor kappa beta. So we just discussed that before with LOX1. And essentially this is an inflammatory cascade and is able to induce the expression of several inflammatory cytokines and chemokines by endothelial cells, such as interleukin-1 alpha, interleukin-8, and chemotactin protein 1. So chemotactin protein 1 is important because this actually attracts the monocytes or the immune cells that get turned into macrophages and converts them into an inflammatory form. And then they continue and say both interleukin-1 alpha and interleukin-8 actively participate in endothelial cell activation, which, while MCP1 is a major chemotactic factor for monocytes and macrophages, therefore endothelial cells can be directly or indirectly activated by tumor necrosis factor alpha. So there's multiple things going on here, but The oxidized LDL activates the endothelial cells, and then it also activates macrophages and mast cells that further release more inflammatory cytokines. So you have the endothelial cell releasing inflammatory signaling molecules, and then you have the immune cells also releasing these inflammatory signaling molecules. And what they're doing is they're causing damage to the actual structure. They're causing more immune cells to move into that area. So it's this kind of feed forward process. It's not a good thing overall. And essentially something to keep in mind here is that the immune cells are coming in to try to deal with this inflammatory stimulus. They're trying to clean up the oxidized LDL to some extent and help to protect the endothelial cells. But essentially with a continuous stimulus, things can kind of get out of hand. So what they further discuss here is, furthermore, as we showed earlier, tumor necrosis factor alpha and histamine have a synergistic effect on endothelial dysfunction and monocyte adhesion. Specifically, histamine applied to tumor necrosis factor alpha activated endothelium significantly increases the surface expression of endothelial E-selectin in a number of firmly adherent monocytes. E-selectin plays a pivotal role in transition from foam cell rolling to crawling and finally to firm adhesion. Due to the increased expression of endothelial E-selectin, the synergistic effect of tumor necrosis factor alpha and histamine on monocyte endothelium adhesion becomes particularly strong under shear flow conditions. So essentially, tumor necrosis factor alpha and histamine are synergistic with each other, and they're helpful to allow the immune cells, the monocytes, to come to the endothelial layer of the arteries and essentially come over the endothelial layer and come into that intimal layer. So if you're not familiar with these different pieces, I discussed the intimal layer and I discussed what was going on in the first episode of this series. So I would refer back to that episode, and again, I will link that into the show notes. But essentially, the oxidized LDL is a key trigger for this entire cascade. Inducing the endothelial dysfunction, activating the immune cells, causing more immune cells to come in. The immune cells then gobble up and eat the oxidized LDL. So let's actually expand this here so you guys can see. And then essentially, once the immune cells gobble up the oxidized LDL, these macrophages or monocytes that can convert into macrophages, they form these foam cells. And the foam cells essentially stay within that atherosclerotic plaque and build up the fatty deposits until they die and they burst, and then you just have these free fatty deposits inside the plaque. So oxidized LDL is a key trigger inside this whole process, and that's why it's so important. So basically what we have so far, this is a brief summary. Normal LDL, the normal cholesterol floating around your blood, what does it do? It transports cholesterol to tissues from the liver. That's a general idea of what it does. There's a bit more, but overall, this is the main piece to know. And then essentially, there's no real negative change to the vasculature from this normal cholesterol. And this is why there's a lot of debate within the cholesterol heart disease hypothesis, because it's not just normal cholesterol that's causing these problems, it's oxidized cholesterol. But oxidized cholesterol is derived from normal cholesterol depending on a multitude of different factors that we'll start to get into in just a bit. Now on the flip side, oxidized LDL. This directly induces vascular damage, as we discussed on the slide with LOX1, and then it also activates the immune cells that can enhance this damaging process. Now what happens here? This can lead to atherosclerosis. So this is the difference between the two types of LDL, and this is going to be important as we build upon this in the subsequent slides. So a new piece that I want to introduce into this overarching picture is what is actually allowing the oxidized LDL or the normal LDL to oxidize and form oxidized LDL. So a key promoter of this is a type of fatty acid, It's called linoleic acid. This is actually an omega-6 polyunsaturated fatty acid. And so I'm going to read a quote here that will describe the process, and then we'll walk through this diagram. So the researchers state here, it was later discovered that oxidation of LDL was initiated by the oxidation of linoleic acid contained within the LDL particles. So if you guys remember, LDL is just a fancy way for the body to carry fats around. And so essentially, the type of fats inside the LDL are really important. So if the fats are really high in linoleic acid, the linoleic acid, and we'll talk about why, is easily oxidizable or peroxidizable, and so then that can lead to the formation of oxidized LDL. So they say indeed linoleic acid is the most common oxidized fatty acid in LDL. Once linoleic acid becomes oxidized in LDL, aldehydes and ketones covalently bind to apolipoprotein B, creating LDL that is no longer recognized by the LDL receptors in the liver, but is now recognized by the scavenger receptors on macrophages, leading to classic foam cell formation atherosclerosis. Hence, the amount of linoleic acid contained in LDL can be seen as the true culprit that initiates the process of oxidized LDL formation, as it is the linoleic acid that is highly susceptible to oxidation. Additionally, increase in the intake of linoleic acid increases the linoleic acid content of very low density lipoproteins and high density lipoproteins, so VLDL and HDL, increasing their susceptibility to oxidize, which further increases the risk of cardiovascular disease. So essentially what happens, let's go over here. So you have an LDL particle, an LDL cholesterol particle. So here's all the little fats, the cholesterol, all different types of lipids. And then you have the protein, apolipoprotein B100. Now, this is a healthy LDL particle. It's able to interact with the LDL receptor. So this is the receptor that allows cells to take fats from this LDL particle, right? Because again, LDL is just transporting these fats around the body. And so the cell, so the cells can take the fats and use them. Now what happens? These different enzymes and then these different metals can oxidize or damage the fats inside the LDL, so you can get this mildly oxidized LDL. Now, the apolipoprotein B100, so the lipoprotein that helps to form and create the function of this LDL particle, it doesn't get oxidized yet, even if some of the fats and lipids are mildly oxidized. So it can still be recognized by this LDL receptor. So that's what you see here, this little red phone looking thing. And then now what happens as the lipids and fats continue to get oxidized, now the LDL, the protein itself, actually becomes damaged from the oxidation products. And now you have a highly oxidized LDL particle. And this essentially does not get recognized by the LDL receptor. Instead, what happens is the immune cells come over and they start to recognize and say, whoa, this is not right. So we're going to engulf this LDL particle so that we can break it down and protect the other tissues from these damaged proteins and these damaged fats. And then essentially the macrophages also release these cytokines here and induce these inflammatory pathways, nuclear factor kappa beta, tumor necrosis factor receptor 1, tumor necrosis factor receptor 2. And essentially, they start to trigger other immune cells to come in and make sure it gobbles up all of this damaged, oxidized LDL to protect the endothelial cells and the other tissues of the body. However, if there's a lot of this going on, the macrophages actually take up too much of this, and you kind of get these obese macrophages that are just loaded with fat. And what happens is they can't handle all of that fat, and eventually they will die in response to all of this fat and their contents. They just have all these peroxidized fats hanging around, and you see this inside the atherosclerotic plaques. So, and these oxidized fats are damaging to the macrophage as well. Peroxidized fats in most of the biological systems are not a good thing. They're quite damaging they can interact with protein structures and other fats and different components inside the cell and create a ton of problems structurally. So essentially the macrophages can only handle so much, and then you see the formation of the foam cell. So this is the general process of the formation of an oxidized LDL and then the interaction with immune cells versus interaction with normal cells trying to take up the fats. And what allows for this oxidative process is loading the LDL with a bunch of linoleic acid and omega-6 polyunsaturated fatty acid. This fatty acid you'll tend to find in soybean oil, safflower oil, sesame oil, a lot of various nuts and seeds. And most of the processed vegetable oils that we're exposed to today, with the exception of some of the tropical oils, and then maybe olive oil and macadamia oil. So next slide here. Let's talk about how polyunsaturated fats are or induce peroxidation or are peroxidized. And this so that we can understand why linoleic acid is causing these problems inside the LDL. So we're going to read this quote here. They say, the carbon atoms that are most susceptible to radical attack are the single bonded carbons between the double bonded carbons of the acyl chains. The hydrogen atoms attached to these carbons are called bisallylic hydrogens. This means that saturated and monounsaturated fatty acyl chains are essentially resistant to peroxidation, while polyunsaturated fatty acids are damaged. Furthermore, the greater degree of polyunsaturation of the polyunsaturated fatty acids, the more prone it is to peroxidative damage. So I know there's kind of a lot of scientific lingo in there. What I'm going to do is I'm just going to show you what we're talking about. So you have a saturated fat here, you have a monounsaturated fat here, and then you have a polyunsaturated fat here. In the saturated fat, you can see essentially these fats are just chains of carbons with hydrogens bonded to them. And then they have an acid group at the end. And so what winds up happening is when these carbons have a double bond in their chains, as you can see here in a monounsaturated fat, you get these hydrogens that have this unpaired element. The carbons aren't fully saturated with hydrogens because this double bond removes some of the electrons available to bind to other hydrogens on this carbon. Now, the problem here is that this double bond and this open area without the hydrogens, I guess an easy way you can think about it is that it allows an area for oxygen and different oxidizing components to come in and attack the fatty acids here and attack these different hydrogens. They're basically not protected because you have a gap in the chain. So the more of these double bonds that you have within the fatty acid, the more likely it is to be attacked by oxidizing components. So what you can see here is in oleic acid, so this is usually found in olive oil and beef fat, it's monounsaturated, meaning it only has one double bond, and the unsaturated component is a reference to the carbons being saturated with hydrogen. So basically what you're seeing is there's one double bond, which means that these carbons are not fully saturated with hydrogen. In the saturated fatty acid, what you're seeing is the carbons are fully saturated with hydrogen. And again, this group is not the main piece we're looking at here. It's these specific carbons bound to the hydrogen. Now, when we come over to linoleic acid, this fat is polyunsaturated. So poly just meaning many. And essentially it has two double bonds. And what happens is this drastically increases the ability of linoleic acid to be attacked by reactive oxygen species and other oxidizing components because these hydrogens there's these gaps here that essentially don't protect the carbon and hydrogen structures at the double bond areas. So it's really kind of like how do we want to put it? It's like if you think of the fat as a fortress or a wall, these are the weak areas of the wall. And the more weak areas you have in the wall, the more likely you are for invaders to break through and damage the wall. And so with polyunsaturated fats, with the double bonds, you have quite a few gaps. Whereas with saturated and monounsaturated, you really don't have that many gaps to cause these different problems. So let's jump to the next one. So, okay, what is the actual difference in peroxidizability or the ability of those fatty acid walls to be damaged? So we're going to read this quote here. They say, Holman empirically determined by measurement of oxygen consumption the relative susceptibilities of the different acyl chains, and you can see the figure over here, of the different acyl chains. Docosahexaenoic acid, DHA, so this is the main fat, one of the main fats in fish oil. The highly polyunsaturated omega-3 PUFA with 6 double bonds is extremely susceptible to peroxidative attack. And keep in mind, it has 6 of those double bonds. So it has 6 weak areas in the wall and is 8 times more prone to peroxidation than linoleic acid, which only has 2 double bonds. So the more double bonds you have, the more likely these fats are to be attacked by peroxidation. So linoleic acid only has 2 double bonds, whereas docosahexaenoic acid or DHA has 6 double bonds, so it's way more likely to be peroxidized. So what they say is DHA is 320 times more susceptible to peroxidation than the monounsaturated fatty acid or monounsaturated oleic acid. So I showed you oleic acid in the previous slide. It only has 1 double bond. So DHA has 6, and it's 320 times more likely to be attacked and damaged from oxidizing components or reactive oxygen species. With the combination of the relative susceptibilities of different fatty acids with the fatty acid composition of membrane lipids, it's possible to calculate a peroxidation index or a measure of the susceptibility of peroxidation for any particular membrane. So what I want to describe here, show you guys here, this is the peroxidation index. So it's how likely a particular fatty acid is to be peroxidized or oxidized or damaged in the face of reaction oxygen species or oxidizing agents. And so saturated fatty acids and monounsaturated fatty acids, they are indeed on the scale here. Just relative to polyunsaturated fatty acids, they are so much less likely to be damaged that they're not even registering. And so what you can see here is you have linoleic acid, and these are both the omega-6 polyunsaturated fatty acids. So you have linoleic acid, and then you have the more unsaturated fatty acid. This fat has more double bonds in it, arachidonic acid. So this one has 4 double bonds, and this one has 2 double bonds. And essentially what you're seeing with these is that the linoleic acid is much more susceptible to peroxidation in monounsaturated fats and saturated fatty acids, and arachidonic acid is even more than linoleic acid. But worse are the omega-3s, surprisingly, considering there's a lot of lip service to their benefit. But linolenic or alpha-linolenic acid has 3 double bonds, and it's not as peroxidizable as DHA, although it is more peroxidizable than linoleic acid. But DHA with 6 double bonds comes in at the most peroxidizable by far. And again, more double bonds, more peroxidation. So here we see 2 double bonds, 3 double bonds, 4 double bonds, and then 6 being the worst here. And then with 1 double bond or no double bonds, the peroxidizability of these fats, at least in comparison to the polyunsaturated fats, is essentially moot. Like they're not even on the same scale at this point. So let's go to the next one. So what actually happens when these polyunsaturated fatty acids are incorporated at the cell membrane or in any of the cellular structures? So I'm going to read this quote here. They say the peroxidation index of a membrane is not the same as its unsaturation index, sometimes called its double bond index, which is a measure of the density of double bonds in the membrane. For example, a membrane bilayer consisting solely of monounsaturated fatty acids will have an unsaturation index of 100 and a peroxidation index of 2.5, while a membrane bilayer consisting of 95% saturated fatty acids and 5% docosahexaenoic acid or DHA will have an unsaturation index of 30 and a peroxidation index of 40. This means that although the 5% DHA containing membrane has only 30% the density of double bonds of the monounsaturated bilayer, it is 16 times more susceptible to peroxidative damage. So essentially, it's not only a count. We're not really measuring the number of double bonds over the total membrane. That's not quite as important as looking at how many double bonds are in the individual fatty acids inside the membrane. And basically, if you have a membrane that has a bunch of fatty acids that are very or polyunsaturated or very polyunsaturated, they have more than 3, 4, or 5 double bonds, that membrane is more peroxidizable than a membrane that's entirely consisting of monounsaturated fatty acids, even though the actual unsaturation index or the amount of double bonds inside the monounsaturated fatty acid membrane is significantly more than the membrane that only has a couple of these highly unsaturated fatty acids. And this is important because essentially the monounsaturated fatty acids inside that membrane, well, they're not really likely to oxidize. You know, the single double bond didn't lead to a high level of peroxidizability. Whereas when you had 6 double bonds, that fat was very reactive, very likely to be damaged by oxidative stress in these different components. So it actually, what you're seeing here is that it's more important to focus on the amount of these highly unsaturated, polyunsaturated fats inside the membrane and overall, and instead of like looking at the total amount of double bonds present in the membrane. So there's a little graphic that goes with this, and it explains some of these things quite nicely. So I want to just go through this a bit. Now, this was in the context of lifespan. This is a AJ Holbert article. I definitely recommend reading some of his research. Very interesting, talking about the membrane pacemaker theory of lifespan. So talking about the unsaturated fat content of membranes and then the interaction of those with lifespan. So what are we seeing here in this graphic? if you have a membrane that's not very polyunsaturated fatty acids, so this here you see membrane fatty acids, and this side is the low membrane polyunsaturated fatty acids. Essentially, the peroxidative susceptibility of that membrane, the ability of that membrane to be attacked by oxidative stress, is quite low. And so essentially, if you have this oxidative stress, which could be, there's mitochondrial production of this, but then there's also many other enzymes or components that can induce peroxidative stress, then you're very unlikely to actually have damage to that membrane. You're not going to have a lot of damage to the DNA inside the cell or the proteins. And what essentially happens, you have slower aging. Now, other things that can impact this are antioxidant defenses. So there are defenses in the cell to manage reactive oxygen species. And then there's also mechanisms to repair these damaged proteins, membrane fatty acids, and DNA. So essentially the recipe here would be to have high antioxidant defenses, solid repair mechanisms, and very low susceptibility to oxidative stress. Then the other thing that you'd want to do is actually minimize or adjust the amount of oxidative stress that you're exposing the cell to overall. Basically, they're talking about here specifically mitochondrial membrane and mitochondrial production of ROS, and that's a whole other story. We'll get into that a little bit. But you want to minimize oxidative stress production to a certain degree. There is some that's necessary. You want to optimize antioxidant defenses. You want to optimize your ability to repair. And then you also want to minimize the ability of the membrane to actually be damaged. So this would be kind of the ideal situation. Now, on the flip side, something that you have that's not as good would be you have a high membrane polyunsaturated fat content. So essentially this membrane is loaded with highly unsaturated fatty acids. And then what happens is the membrane is very likely to be damaged by oxidative stress. It'll damage DNA, it'll damage the proteins. And then if antioxidant defenses are great and repair mechanisms are good, that's also helpful. But if ROS is kept to a minimum, that also would be helpful. But overall, you have a higher susceptibility to that oxidative stress overall. And so with a lot of unsaturated fats in the membrane. So anytime you adjust any of these other dynamics, you're going to start to see very significant damage to the cellular structures, because essentially you can think of, am I going to build my house out of cement over here, or am I going to build my house out of paper? And so tons of unsaturated fatty acids is the equivalent of building the house out of paper. So yeah, if you have a house built out of paper and there's no hurricanes and there's no earthquakes and there's not a lot of rain and perhaps the climate doesn't get too hot, yeah, your house will stand. But if you have any adjustment to any of those things, the house is going to fold. Or in this case, the cell is going to fold. But if you have your house made out of cement, even if you have the earthquake, even if you have a hurricane, even if you have some rain, even if you have higher extremes in temperatures, the structure is going to be stable. And so this is what we're kind of looking at here with the polyunsaturated fats. And this also applies to what's going on inside the LDL cholesterol. So that's why I went through this, even though it seemingly seems a bit tangential. But essentially, if we're building the cholesterol out of tons of polyunsaturated fats, then we're more susceptible to oxidative stress and damage. And that's essentially the main key point to get through here. So let's talk about what this looks like. So a normal LDL cholesterol plus adding a bunch of omega-6 polyunsaturated fatty acids heavily increases the risk of forming oxidized LDL. So this is the basic overview of these last few points that I just discussed. Now, we're going to add another layer to this, and we're going to talk about, okay, we can think of the omega-6 kind of like dynamite. We're loading our cells up with dynamite, or we're making our cells, the structure of our cells or the structure of the cholesterol, quite weak, or the cholesterol is kind of loaded with this very reactive dynamite. But it's not only loading it with this component that's problematic. Essentially, when you have to have some type of stress on the system or production of reactive oxygen species to light that dynamite to a large extent. So essentially, there's two components here, and hence the title of this presentation. But the reactive oxygen species, you can think of the fuse, and the polyunsaturated fatty acids, you can think of the dynamite. And so I'm going to read this quote here, and we'll jump into this. So oxidative stress manifests itself in excessive reactive oxygen species generation and subsequent oxidation of LDL particles. In pathologic or stress conditions, reactive oxygen species overwhelms antioxidant systems, leading to an imbalance, which in turn causes oxidative stress and irreversible changes in cell compounds, including proteins, carbohydrates, and lipids, in addition to being able to disrupt normal cellular signaling mechanisms. So essentially, if you have too much ROS, it can lead to damage to the actual structures. So you can think of the ROS as kind of like that hurricane, that earthquake, that change in weather that is able to damage either the cement or the paper house. And you can think of the polyunsaturated fatty acids as the polyunsaturated fatty acids versus saturated or monosaturated as the building blocks of that house. So when we build our house out of the paper and then we have these other factors come in, it damages all of the structure. But when we build our house out of the cement or which would be the polyunsaturated fatty acids or saturated fatty acids, even if we have these different components, these reactive oxygen species interacting with the cell, what we find is that the cell or the house doesn't fold very quickly. And so essentially, this is another diagram here breaking down this concept. And so what you see is you have different components that can increase oxidative stress, and we're going to talk about some more. But cigarette smoke, radiation, ultraviolet light, heavy metals, air pollution, there's a couple other things we'll talk about. These create these reactive oxygen species, and we'll get into these in just a second. And then essentially, all of this can be prevented by the antioxidant defense systems. So essentially, you have you have different vitamins, vitamin C, vitamin E, glutathione, and then different enzymes, catalase, superoxide dismutase, glutathione, the glutathione enzyme family. And so essentially, that can help protect against these reactive oxygen species production. And the other thing I want to mention here is there is another component that can create reactive oxygen species, and that is endogenous components. So that's the mitochondria can produce them under certain circumstances. Issues inside the endoplasmic reticulum of the cell, kind of like the factory area of the cell. It produces multiple different products, packages them, ships them, exports them. Kind of think of it like an Amazon warehouse. If there's problems in there, you can get reactive oxygen species stress. Peroxisomes. So this is an area where fats are oxidized. Different enzymes used for defense. So NADPH oxidase, usually used in immune defense, and then different inflammatory enzymes like oxygenase and cyclooxygenase. So all of these also create oxidative stress in conjunction with these external or exogenous factors. Now, what you see here on this spectrum is that less oxidative stress leads to less issues with disease processes or pathogenesis. So cardiovascular disease, cancers, diabetes, neurodegenerative disorders are all characterized by high amounts of oxidative stress, and essentially oxidative stress where the body is unable to actually deal with that oxidative stress. The antioxidant defense mechanisms are not strong enough to handle this, and you start to get severe dysfunction. The other thing that they don't mention on here, though, with this is, and it's the component that we're adding into the mixture, is that the cellular structure, or in this case, perhaps the LDL, the cholesterol structure, if it's made out of polyunsaturated fatty acids, all of these different components are more likely to create oxidized LDL because of the ROX production, and the ROS can actually damage the polyunsaturated fatty acids inside the LDL. So there's multiple pieces that we're working through here. We have exogenous and endogenous sources of reactive oxygen species. We have systems, again, that can manage these reactive oxygen species. And then essentially we also have how likely are the cellular structures to be damaged by the reactive oxygen species. And that's dependent upon, to a large extent, what types of fatty acids are contained within these different structures inside the cells, tissues, and the body at large. And then essentially a modulating effect where if we have too much oxidative stress, we lead to disease processes, whereas less, there's always going to be a degree of some. So we're not going to actually absolute zero. But if we have a less or amount less than a threshold that's producing disease, up to a certain point, it's probably better to keep it lower overall. So this is a general overview of oxidative stress. Now, what are reactive oxygen species? So we're talking about oxidative stress, we're talking about reactive oxygen species, but essentially what's going on here? So here's a definition. And so reactive oxygen species is also often called a free radical. So they say a free radical is any molecule capable of independent existence that contains one or more unpaired electrons. ROS is a collective term that includes oxygen radicals as well as non-radical derivatives of oxygen. So here we have some ROS structures, and what we can see, and not necessarily in the oxygen structure, but in the superoxide anion, peroxide, hydrogen, or the hydroxyl and hydroxyl ion, what we can see is that on these different, this is oxygen, and these little dots are the electrons. There's an electron that's unpaired, and so essentially, what's happening, this is general chemistry ideas, is within the oxygen's valence shell, or this basically these orbiting electrons, they want to have a complete orbit. So basically there needs to be a certain number of electrons orbiting the oxygen molecule, and if there's not, then the oxygen will go out and will try to actually get an electron to add to this orbital layer to make sure that it's stable, to make sure that it's full, so you don't have these different negative charges. Now with oxygen, hydrogen peroxide, these can actually split relatively easily and create these situations where you have the unpaired electrons. So they are very reactive, very easily split to create these different radicals and ions. So yeah, this is the general idea behind the ROS, and basically these species can be produced by multiple different mechanisms. So we're gonna dive into those right now. So what are the major producers of reactive oxygen species? So I'm going to read this quote here, and then we'll go through the diagram. Reactive oxygen species can be generated at various sites and under a variety of conditions. These include the membrane NADPH oxidase of polymorphonuclear leukocytes, so these are immune cells, during ischemia reperfusion, as well as other enzymatic reactions, so lipoxygenases, cyclooxygenases, peroxidases, and other heme proteins. The enzyme xanthine oxidase, peroxisomes, or hepatic P450 microsomal detoxifying systems. In healthy tissues under normal conditions, most ROS are generated by the mitochondrial respiratory chain. So essentially, we have a variety of enzymes here that can create reactive oxygen species. And then you also have the mitochondria, basically where the cell is producing energy, where you can also get the production of these reactive oxygen species. So here's this is a nice diagram kind of covering this. Now, I would ignore or I guess we can incorporate the xenobiotics and microbes, but essentially the endogenous systems, as we kind of discussed, so we have these different enzyme systems that can produce that can produce reactive oxygen species. We have the peroxisomes, we have the mitochondria, we have the endoplasmic reticulum stress, kind of what I talked about before. Then you also have the inflammatory mediators that will induce it, radiation, and then you also have toxins and infective components that can also induce ROS. So there's multiple components that can drive ROS. Obviously, there's actually a lot of components that can drive ROS. So why is this actually important in the context of oxidized LDL? Well, essentially, if you're loading your LDL up with polyunsaturated fatty acids like linoleic acid, there's tons of different components or different states that can produce large amounts of reactive oxygen species, and that can essentially damage the linoleic acid and lead to the formation of oxidized LDL. And again, this is very important because that oxidized LDL, as we laid out in the beginning, is actually causally involved in the production of atherosclerosis. So we have these two really important components, and the reactive oxygen species component can be derived from multiple different areas. So essentially, the reason I'm going into this in such depth with the reactive oxygen species is there's not really a good argument to say, well, if we keep ROS low, then we can, then some polyunsaturated fatty acids inside the LDL or inside the cellular structures are a good idea. And the reason why is because there's a lot of elements of life that will increase ROS. And so it's almost impossible to always keep ROS low. So you want to make sure that when you're exposed to ROS, you're not damaging all of the cellular structures. So basically, this brings us down to a general synopsis, or the next level synopsis or summarization of what we discussed so far. So we have normal LDL cholesterol. If we load it with a bunch of omega-6 polyunsaturated fatty acids, and then we expose it to reactive oxygen species, well, now we get increased oxidized LDL formation. And why is this important again? Because that increased oxidized LDL formation is what is driving the heart disease process causally, not associatively, causally, and the mechanisms have been laid out. Okay, so now what we're going to talk about is what are all of the different, or not all, but what is a good sampling of different things that can directly induce reactive oxygen species inside the body. And again, the reason I'm laying this out is because I don't think there's a good argument to say that, oh, if we have some polyunsaturated fatty acids inside the cells or tissues, if we keep ROS low, then we should be fine. Essentially, what I'm going to get to here is that it's like there's many situations that we'll be exposed to in life that will drive ROS and oxidative stress. So the first one is just viral infections. If you get a cold, if you get COVID, if you get any type of stomach virus, you're going to drive a lot of ROS production. And basically, this is just describing some of the different mechanisms. So here's the quote: increased mitochondrial reactive oxygen species production resulting from direct interactions between viral proteins and mitochondrial membranes and leading to respiratory dysfunction has been abundantly reported. In addition, viral interactions with other mitochondrial associated components could also result in mitochondrial ROS production. So this component here is just looking at different viruses. So hepatitis C, hepatitis B virus, enteroviruses, HIV. They all have different proteins that can. This is the mitochondrial membrane. You can see the different mitochondrial complexes, and essentially the proteins from these viral components can all interact with multiple areas inside the mitochondria and induce reactive oxygen species stress. And so essentially, if you are exposed to any of these infections, you're going to have a higher amount of reactive oxygen species production. And so most people get a cold once or twice a year or a stomach bug or something along those lines. So this is a relatively regular occurrence for a lot of people. And again, this is a source of reactive oxygen species stress directly. Also, some of these proteins can interact with the endoplasmic reticulum and create ROS there. So it's not just the mitochondria. There's multiple different components. But I wanted to show viral infections are going to increase reactive oxygen species stress. And most of us experience these on a regular basis. So again, we don't really want to, in the face of this, the goal would not to have our cholesterol components or our cellular components it can load it up with higher amounts of polyunsaturated fatty acids. So another component that can increase oxidative stress is, surprise, surprise, endotoxin. And so what I'm going to do here is I'm going to read this quote and then we'll get into the image. So as central components of the respiratory burst and activated leukocytes, the reactive oxygen species and reactive nitrogen species play an essential role in the host immune defense against pathogens. However, based on their nonspecific and highly reactive nature, radical and non-radical products of redox metabolism may be potentially harmful to host cells. Apart from virulence factors expressed by pathogenic microorganisms, e.g. microbial cytotoxins such as endotoxin, the excessive production of ROS and reactive nitrogen species by activated immune cells creates a highly cytotoxic milieu that contributes to the direct damage of target organs in the context of unbalanced inflammatory responses. Okay, so if our cells or our cholesterol particles floating around in our blood are exposed to large amounts of ROS, again, then we're likely to oxidize these components if they're filled with polyunsaturated fatty acids. Now, microbial components, so microbial toxins, induce immune cells to produce reactive oxygen species as a defense mechanism. However, what we're seeing here is the reactive oxygen species can actually damage the host or the organism's body in and of itself, and then also the production of these immune mediators will, as we talked about previously, also further induce ROS production. So if we're in this circumstance where we're exposed to, and we'll go through this image here, if we're exposed to, and we're all exposed to this, any type of gut-derived bacterial products, they'll go into the liver, they'll get into the liver immune cells. They're called Kupffer cells, and also into circulation. And essentially, these bacterial-derived products activate immune cells, And then the immune cells create these reactive oxygen species, and then they also produce these inflammatory mediators like we talked about before, tumor necrosis factor alpha, and this drives the reactive oxygen species. So if our liver, if our cells, if our cholesterol particles are loaded with polyunsaturated fatty acids, what you can essentially be seeing is in the state of infection or chronic low-level endotoxemia, you essentially are in a very delicate situation if your tissues are loaded with polyunsaturated fatty acids, because these fatty acids, again, very likely to oxidize, as we described earlier, and now you have a state that drives oxidative stress. And this one's quite important because most of us on a regular basis are chronically exposed in varying amounts on the spectrum to gut-derived endotoxin and gut-derived components from different microbes. Again, every time we put food into the digestive tract, as largely once it gets into the colon, but for some people perhaps in the small intestine, you're going to have these different microbial components being produced, and a solid intestinal barrier helps protect against them. But if you're under stress, if you have a viral infection, if you've just heavily exercised, you'll find that you may have some minor increases in gut permeability, and that can allow these components in, and they can drive this reactive oxygen species. And there are some theories around gut-derived endotoxin as a key component inside multiple different disease processes, whether that's hypogonadism or lower testosterone in men, or dysfunction in thyroid profile, or also dysfunction leading to diabetes or obesity, metabolic syndrome, heart disease, etc. So we don't want to have both factors present. Ideally, we want to minimize both factors. And those two factors are, again, highly reactive fatty acids, and then the reactive oxygen species that can ignite the damage to those fatty acids. So another component where we'll see this is exercise. General exercise, both aerobic and also anaerobic, so like bodybuilding, lifting, as well as going for runs or playing different sports, does induce oxidative stress, and this is well known. So what they go to say here is they say, thus it has been demonstrated that exercise-induced muscle damage causes different time course changes in general oxidative stress responses, mainly due to reactive oxygen species and free radical production centered on phagocytes. Several prior studies have reported that free radical levels in blood and skeletal muscle increase immediately in exercise without muscle damage, which is in stark contrast to eccentric contraction exercise, in which an increase in free radical levels in blood is not observed until 72 hours later. So essentially, in any type of like lifting exercise, the concentric action, so you can kind of think of a bicep curl. If I'm actually curling, that's a concentric action. And as I let the weight down, that's eccentric. The concentric action will induce immediate reactive oxygen species, the concentric one, whereas the eccentric contraction will actually induce delayed production of free radicals, reactive oxygen species, about 72 hours later. That can be what people experience as DOMS, or delayed onset muscle soreness, or that's one of the theories behind it at least. And so the next one here, they say studies conducted since the 1990s have reported that acute aerobic exercise increases the levels of markers of oxidative damage of lipids, proteins, and DNA. Similarly, many studies have reported that acute aerobic exercise affects antioxidant levels and redox balance. So we're just going to go over here. Again, I have a nice diagram for us. But acute exercise inside the skeletal muscle will drive mitochondrial produced oxidants, xanthine oxidase, so one of these enzymes, NADPH, another enzyme, and then the immune cell production of oxidants, so reactive oxygen species. This contributes to oxidative stress. Now, what determines how well you deal with that oxidative stress is actually the intensity of the exercise. So how strong is that exercise stimulus? Your nutritional intake. So do you have the micronutrients? Do you have a lowered amount of polyunsaturated fatty acids? Do you have the adequate protein, carbs, and fats to deal with this stimulus and not upregulate the stress hormones? Is your body adapted to dealing with this stress? Have you been training on a consistent basis? How much oxidative stress do you have at rest? Are you loaded with polyunsaturated fatty acids in your tissues? Are you a smoker? Do you have excessive stress levels overall, which can upregulate stress hormones and drive oxidative stress? And then also, what is your age? Are you dehydrated? there's differences between men and women, and then hypoxia, which this would be maybe perhaps differences in altitude training or not. These will all adjust what's going on with your ability to manage that oxidative stress. So exercise is another component that drives oxidative stress. And again, this is a component of most people's lives on a consistent basis. So we have infections, we have gut-derived endotoxin products, then we also have exercise as things that can increase oxidative stress. Next one is stress hormones. So there's a couple stress hormones. So glucocorticoids, or you can think of here as cortisol, can actually directly induce reactive oxygen species at the vasculature. So if you're under heavy chronic stress, you have elevated cortisol levels. This can be driving an increase in reactive oxygen species and oxidative stress, which if your cholesterol and your cells are loaded with polyunsaturated fats, the idea here would be that you'd be more susceptible to the negative impact of this because those fats are likely to be damaged by their reactive oxygen species stress. So essentially what we see here is vascular endothelial cells are also capable of producing reactive oxygen species such as superoxide, hydroxyl radicals, and hydrogen peroxide generated from various enzymatic sources. Because superoxide reacts with nitric oxide more promptly than with superoxide dismutase, intracellular overproduction of superoxide results in reduced nitric oxide availability through enhanced elimination of nitric oxide. Thus, we hypothesize that glucocorticoid excess may elicit the overproduction of reactive oxygen species from endothelial cells and thereby reduce nitric oxide availability to cause an impairment of relaxing activity of vascular smooth muscle cells. So essentially, nitric oxide is needed for a relaxing effect on the endothelial cells, and too much ROS impairs that ability. And so the researchers are testing here to do an excess of glucocorticoids, of cortisol, or an excess production actually lead to increased ROS, and then they're trying to see the effects on nitric oxide. So basically what they said is our study using several inhibitors demonstrated that oxypurinol, quinacrine, DPI, DIDS, CCCP, and TTFA inhibited. So DEX is dexamethasone. It's a synthetic glucocorticoid, induced reactive oxygen species production to almost the same extent. Based on these findings, we can consider that mitochondrial electron transport chain, xanthine oxidase, and NADPH oxidase are major sources of glucocorticoid-induced reactive oxygen species production in human umbilical vein endothelial cells. So they're testing this in a cell model, looking at endothelial cells and seeing how they respond to glucocorticoids. And essentially, these different components are all inhibitors of those various enzyme systems that we discussed previously in term overall. So basically what they're seeing is if they inhibit these various enzyme systems and they expose the cells to dexamethasone, the synthetic glucocorticoid, is there still ROS production? And what they're seeing is that when they inhibit these different enzymes, there's actually not ROS production. So dexamethasone must be increasing ROS by affecting these different enzymes. They further on go here to say that we showed that dexamethasone-induced ROS production was effectively suppressed by an uncoupling agent of oxidative phosphorylation that can abolish mitochondrial transmembrane potential, as well as inhibitors of complex 1 and 2. These results suggest that glucocorticoid-induced ROS overproduction is mainly derived from proton electrochemical gradient generated in mitochondrial electron transport chain. So essentially what they're showing here is that glucocorticoids are mainly inducing the ROS overproduction at the endothelial cells by adjusting metabolism. Now, this is actually really interesting because it goes directly into the bioenergetic view and as far as viewing energy metabolism as central to all the different pathologies or the variety of different pathologies that you can be exposed to. But essentially what you're seeing here is glucocorticoids, or in this one's a synthetic one, can drive ROS production at the endothelial cells. And this is very specifically important because ROS production at the endothelial cells is where you're seeing issues with LDL oxidation. And there are associations with glucocorticoids and cardiovascular disease. I'm not going to discuss them just for the sake of time inside this lecture, but they are, they are very present within the literature. And basically this graphic here is just showing us the effects of glucocorticoids. So basically the glucocorticoids have a negative effect on nitric oxide. They induce angiotensin II and endothelin 1. We'll talk about angiotensin II in just a second. And then they can also cause issues around cell death, expression of pro-inflammatory molecules at the endothelial cells, and they can affect cell proliferation, viability, and cell migration. So these are the proliferation of the endothelial cells and their viability, and then also the migration of the immune cells overall. So higher levels or chronic exposure to glucocorticoids actually have quite a negative impact overall on the vascular system over time, and it's through multiple mechanisms, but a decent portion of this is around reactive oxygen species production at the endothelial cells. Next one here is catecholamines. So essentially what we're seeing here is catecholamines do very similar things as the variety of the different stress hormones. Higher levels of the catecholamines essentially can increase the production of reactive oxygen species. And again, this is not a good thing in the face of having your cholesterol particles or your cellular structures loaded with polyunsaturated fatty acids. So what they say here, I'm probably going to read this first one and not the second one per se. We'll see. They say stimulation of alpha-1 adrenergic receptors by blood-borne norepinephrine or by increased release of norepinephrine from vascular nerves induced by injury is proposed to activate NADPH oxidase-dependent generation of this reactive oxygen species, and in turn, the hydroxyl radical. Hydrogen peroxide causes activation of the EGFR ERK1/2 pathway and subsequent induction of protein synthesis and proliferation. Although identification of the steps that are proximal and distal to indicated mediators in this pathway await investigation, the pathway suggests a direct link or risk factor status may exist between catecholamines injury and progression of vascular disease. So essentially, the catecholamines, they can increase the activation of one of the enzymes we discussed, NADPH oxidase, and produce these different ROS. And then essentially the ROS can lead to the production. So there's enzymes that will convert this into these reactive oxygen species to hydrogen peroxide, and then that can cause activation of these different inflammatory molecules, and then that's directly involved with the progression of vascular disease. So you're seeing the catecholamines can also drive ROS production at the vascular level, and again, this is directly involved with having these cells and the cholesterol particles loaded with polyunsaturated fatty acids, which would increase the likelihood of those polyunsaturated fatty acids to oxidize. And so what's the overall importance and context here? Well, if you're under stress, and you're upregulating catecholamines and you're upregulating glucocorticoids, what you can see an increase in that ROS. So there's no point in time where you're throughout life where you're not going to have different periods where you'll have stress and you'll upregulate these adaptive hormones. They're needed for these stressful situations in many circumstances. And so under those circumstances, you're going to drive ROS. It'd be important to not have the structures of your cell be liable to be heavily damaged by that ROS, aka having those structures loaded with polyunsaturated fatty acids. And then in this context, also these things could drive ROS production that can possibly lead to the increased oxidation of the cholesterol particles or even these polyunsaturated fatty acids inside the different cellular components. As we saw with the glucocorticoids, they're actually driving increased mitochondrial ROS production. So if the mitochondria are membranes are loaded with polyunsaturated fatty acids and you get more ROS overall, you can possibly see an increased damage to the mitochondrial membrane. So again, basically all I'm trying to show here is that under different stressful circumstances, whether that's psychological stress, whether that's energetic stress, whether that's exercise-derived stress, you can find that you'll increase ROS overall through multiple mechanisms, and then you don't want to be doing that if your tissues are loaded with polyunsaturated fatty acids that are very liable to this oxidation. The last two here are speaking, so angiotensin II, and then the next one is parathyroid hormone, are going to speak to mineral balance overall and fluid balance. So essentially in states where you don't have adequate mineral balance, or you don't have adequate fluid and electrolyte balance, you can increase these different mediators, angiotensin II and also parathyroid hormone, and they also directly drive reactive oxygen species stress at the vasculature, and they also drive the atherosclerotic process. So overall, this is adding another circumstance. If your calcium, vitamin D, magnesium, vitamin A, vitamin K intake isn't sufficient, you'll drive PTH. And then for angiotensin here, if your sodium, potassium, and fluid intake and your overall blood pressure is not sufficient, then you'll drive the production of angiotensin. And angiotensin is also driving this oxidative stress that we see here. So what they say here is in the vascular system, reactive oxygen species are produced in endothelial, adventitial, and smooth muscle cells primarily by NADPH oxidase, which produces this reactive oxygen species radical upon being stimulated by angiotensin II, endothelin 1, and neurotensin 2, among others. Additionally, increased mechanical forces due to elevated blood pressure, which these different components induce, such as unidirectional laminar and oscillatory shear stress, can contribute to increased reactive oxygen species production. So essentially, the increased blood pressure can adjust the flow of blood throughout the arteries, and then that flow can, based on if it has like an oscillatory shear stress, can increase ROS production. I discussed oscillatory shear stress inside my previous presentation on cardiovascular disease, so we can get in. If you want to check out what exactly this is, you can go and do so in that presentation. The next piece that they discuss here is nitric oxide signaling plays a role in endothelial-independent vasorelaxation, which is mainly mediated by nitric oxide. Endothelial cells release nitric oxide, thereby causing vascular relaxation. NO is rapidly degraded by oxygen-derived free radical. Superoxide anions produced by nitric oxides synthases react with nitric oxide to produce, these are NADPH oxidase, react with nitric oxide to produce peroxynitrate, thereby decreasing the bioavailability of nitric oxide and inducing vasoconstriction. Thereby, hypertension is associated with the reduced availability of NO and increased oxidative stress. Angiotensin II induced hypertension involves the activation of redox-dependent signaling cascades and NADPH oxidase-induced generation of ROS. Angiotensin II, the major bioactive peptide of renin-angiotensin system, plays an important role in many vascular processes such as vasoconstriction, hypertrophy, fibrosis, inflammation, and aging. Vascular NADPH oxidases are all regulated by angiotensin II and NADPH oxidized derived reactive oxygen species signaling mainly mediate the angiotensin II effects. Angiotensin II activates the angiotensin 1 receptor, which increases the production of the superoxide this reactive oxygen species radical via two membrane-bound NADPH driven oxidases. So I know that's a lot there, but essentially what you're seeing here is that angiotensin II, which is involved in the RAS system and around regulation of fluid balance, sodium and potassium balance, and then blood pressure, under circumstances where perhaps there's an imbalance with the electrolytes or imbalance in fluids, will drive this angiotensin production. And then essentially what angiotensin does is it increases reactive oxygen species directly at the vasculature, which again, if you have LDL loaded with polyunsaturated fatty acids like linoleic acid, and you have a high exposure to angiotensin II for whatever the different reasons are, now you have an agent that creates reactive oxygen species, and you have a bunch of fatty acids that are easily oxidized in the face of reactive oxygen species. So again, recipe for disaster. And the overarching picture, we're looking at all the circumstances that you can be exposed to that will drive ROS, and essentially there's like so many varied circumstances that many people are exposed to regularly, where it just doesn't make sense to load the tissues up with those the polyunsaturated fatty acids. Now the last one is PTH, and again, PTH is a hormone that regulates calcium balance. So if you don't have enough calcium, vitamin D3, or any of these components, then you can also drive oxidative stress, particularly at the vasculature. And this is another important piece overall for the same things that I've been kind of reiterating in terms of having your cholesterol particles or the cellular components loaded with linoleic acid. So basically what we're seeing here is indeed PTH has been directly implicated in a wide range of vascular alterations such as endothelial dysfunction, vascular calcification, and vascular remodeling, which lead to atherogenesis and arterial stiffness. The relative contribution of vascular alterations in PTH-induced increased cardiovascular disease is still debated, but mounting evidence supports the pivotal role of endothelial dysfunction even in early phases of chronic exposure to higher PTH levels. Altogether, our data represent the first direct evidence that PTH causes endothelial dysfunction in ROS-dependent manner through a surge of mitochondrial calcium 2+, or calcium ions. This oxidative response leads to the impairment of selected signaling transduction pathways with altered nitric oxide production, impaired endothelial phenotypes such as angiogenesis. So essentially, what we're seeing here is parathyroid hormone released by the parathyroid glands interacts with the vascular cells through the parathyroid receptors, and there's multiple signaling pathways, but you can see this increase in calcium ions that drive oxidative stress through the mitochondria, and then this oxidative stress can lead to vascular inflammation, vascular calcification, hypertension, cardiac hypertrophy, etc. So essentially, it's the parathyroid hormone, another component, is driving this cardiovascular disease process, this atherosclerotic process, through oxidative stress. So there's multiple components here. You have endotoxin, immune activation, you have infection, viral or bacterial, you have exercise, you have nutrient deficiencies from a calcium perspective, so calcium, magnesium, vitamin D3, you have any changes with blood pressure or fluid status or sodium potassium ratios. All of these different components can drive ROS. So they're so varied and they're so ubiquitous in the environment that if you're going to be exposed to this reactive oxygen species stress, and then again, those polyunsaturated fatty acids are going to increase the likelihood of damage under these circumstances. Now, the last couple ones that I have to discuss here for the oxidative stress component are pollutants. So different toxins, heavy metals, all these different components can drive oxidative stress. This specific slide is talking about particulate matter, or different particles that you can inhale that also drive oxidative stress at multiple different levels. And basically what they say, the three main hypotheses of how inhaled particles could cause cardiovascular effects. Particles induce an inflammatory response in the lungs, leading to the release of cytokines and other mediators that spill over into systemic circulation. So again, these cytokines and cell mediators can drive ROS directly at the vasculature as well and oxidize the LDL. Then it says ultrafine particles themselves may translocate across the alveolar wall and directly interact with the cardiovascular system. This process may be aided by inflammatory cells. So certain particles that you could inhale, different pollutants, can move across the alveolar wall. So the alveoli are the little sacs in the lungs that help with air, carbon dioxide and oxygen exchange. And when they get into the vascular system, they can drive an immune and inflammatory response. And then they also talk about the particles can activate autonomic nervous system through sensory receptors on the alveolar surface, perhaps representing a rapid mechanism by which particles can affect cardiovascular function. Oxidative stress could play a role in exacerbating numerous stages of each pathway, as well as promoting interactions between pathways. So oxidative stress is a key component in multiple of these pathways. But interestingly, what they're saying is that the particles can activate the autonomic nervous system. So the main mediators of the autonomic nervous system are the catecholamines, such as epinephrine and norepinephrine, which we previously discussed can directly drive oxidative stress at the vasculature. And then basically what we're seeing here in this graphic is there's through multiple different steps, you're seeing that these airborne particulate matter, there's multiple different types, can drive oxidative stress throughout the body. And this has been linked directly to cardiovascular disease. And it's through these oxidative stress mechanisms. So again, we have this interplay of oxidative stress. If you're in an environment where you're exposed to particulate matter, probably be a good idea to minimize the amount of linoleic acid consumption that you're loading into your tissues and your cholesterol particles on a regular basis. Last one is last two, metabolic dysfunction. So this one's actually gets directly into the bioenergetic sphere ideas around the importance of having metabolism function appropriately. And it's going to talk about a very key piece here that's a bit tangential, but I wanted to jump into just a bit. So what the researchers say here is they say healthy cardiomyocytes use their energy sources flexibly to match ATP demands and oxygen availability. Fatty acid beta oxidation, which mainly takes place in the mitochondria and peroxisome, requires high oxygen availability and is inefficient in times of high ATP needs. In diabetic hearts, due to high circulating and stored fatty acids, fatty acid oxidation continues to be a predominant source of ATP, and there's a relative loss in energy contribution from carbohydrates and glucose during periods of high ATP demand. The loss in flexibility between energy sources causes reduced cardiac efficiency and contractile dysfunction, which is a hallmark of diabetic cardiomyopathy. So this is actually quite interesting because essentially, despite all the lip service in the low carb sphere to metabolic flexibility being a function of eating a low carb diet, what they're showing here is that diabetic hearts actually oxidize fatty acids more so than carbohydrates compared to healthy hearts, and that loss of ability to oxidize carbohydrates effectively during periods of high demand leads to a decrease in cardiac efficiency or production of ATP, and then leads to dysfunction and contractility of the cardiac cells because they don't have enough ATP. So in this circumstance, too much fatty acid oxidation is actually not good for the heart. Now, what they say is an increase in fatty acid oxidation causes increase in mitochondrial intermembrane potential and stimulates reactive oxygen species, which further causes oxidative damage of mitochondrial proteins and DNA, making them less efficient in energy metabolism. Mitochondria in diabetic cardiomyopathy are further compromised due to chronic upregulation of uncoupling proteins found within the inner mitochondrial membrane that adversely affect mitochondrial energetics and calcium stores. The cumulative damage to mitochondria caused by metabolic derangements and diabetes further upregulates ROS production that promotes apoptosis as suggested by animal studies. Over time, apoptotic cardiomyocytes are replaced by fibroblasts, or essentially they're turned into fibrotic tissue, and this change in tissue composition has been implicated in heart failure with preserved ejection fraction. So what's going on? When we have metabolic dysfunction, which is characterized by excess fatty acid oxidation with an inability to oxidize carbohydrates effectively, you get a disruption in the production of ATP, but that excess fatty acid oxidation drives reactive oxygen species production. If you want to understand the exact mechanisms of how this is actually occurring, I described this in my Randle Cycle video on my channel, so you guys can check that out. I'll put the link in the description. And essentially, what you start to see is the mitochondria start to produce tons of ROS that leads to oxidative stress, and that oxidative stress can eventually lead to cell death. And then when the cell dies, it's just turned to fibrotic tissue, non-functional tissue, essentially. So this is also quite important because this what eventually leads to the cell death and adjusts the cardiac cell's response to the oxidative stress, particularly in the mitochondria, is the unsaturation of the mitochondrial membranes. And then if you're having a high amount of oxidative stress overall at the cardiac cells or the vasculature or whatnot, then that can also expose the different lipoproteins like the LDL and HDL to reactive oxygen species. And what you tend to see inside diabetes is increased reactive oxygen species stress. And then you can see increases in oxidation of the LDL and different components, which can further drive the atherosclerotic process. And within diabetes, you tend to see an increased risk and an increased prevalence overall of cardiovascular disease. And again, the diabetic state is characterized by this metabolic dysfunction on the cell, but this excess fatty acid oxidation and metabolic dysfunction of the cell in diabetes creates a state of high amount of reactive oxygen species or a high level of oxidative stress, which can affect numerous other processes, including the oxidation of LDL particles, which again, I think that's one of the main mechanisms is why you're seeing this increased atherosclerosis and heart disease inside diabetic people with diabetes. And so essentially what I want to show here is that if you're in any states of metabolic dysfunction, you're also increasing your level of oxidative stress. So again, another piece to the picture to show that the oxidative stress is prevalent in many circumstances. And so you're not going to want to create a situation where you're more susceptible to the damaging effects of this oxidative stress by loading up LDL particles and tissues with polyunsaturated fatty acids. Last one, I'm pretty sure this is the last. Oh no, two. I keep saying the last one. I put way too many different options on here. Basically, I wanted to get through all of the different possibilities or a large amount of different possibilities that can drive oxidative stress. But this one is nutrient deficiencies. And essentially, I didn't go through all of the different nutrients. There's vitamin D, vitamin E, vitamin C are some of the main ones that people think about, vitamin A, but I just chose zinc specifically as like a basic mineral that's heavily involved with the antioxidant systems, and where a deficiency of it can drive oxidative stress, and specifically related to vascular and cardiac dysfunction, which can drive cardiovascular disease. So what the researchers say is to say, since zinc is not redox active, so it's not directly involved itself in oxidative reduction reactions, the antioxidant function of zinc is through some indirect mechanism of action. In mammalian cells, the main redox active metals, including copper and iron, catalyze the production of reactive oxygen and reactive nitrogen in species, which are capable of oxidizing cellular components such as lipid bilayers. So lipid bilayers is basically the cell membranes, which are composed of these different fats. Zinc competes with these redox active metals for negative charges in the lipid bilayer, and as such, it protects cell membrane from lipid oxidation. Zinc is also able to interact with thiol or sulfohydryl groups in proteins and peptides to reduce the reactivity of the sulfohydryl groups. This stabilization prevents intramolecular disulfide formation, and thus zinc functions as an antioxidant to protect these molecules from oxidation. So having adequate zinc on board protects the proteins from interacting with different components and forming these disulfide formations via the sulfohydryl groups inside the different proteins. And then it also protects the fats inside the cell membrane from lipid oxidation, and it can, by interacting or competing with copper and iron, which actually drive oxidative stress. Then zinc additionally serves as a cofactor in antioxidant enzymes such as superoxide dismutase that scavenges superoxide ions. Although zinc is tightly incorporated in superoxide dismutase 1, is unlikely spontaneously released from the protein. Just because of changes in zinc status, there has been evidence to show that zinc supplementation affected superoxide dismutase protein stability and protected mice bearing unstable superoxide mutant genes. Upon zinc depletion, superoxide dismutase 1 mutants underwent conformational changes due to zinc dissociation, which resulted in oxidative stress and endoplasmic reticulum stress. So essentially, zinc is also involved in some of the major enzymes that help to clean up oxidative stress. So this enzyme is superoxide dismutase. And so this graphic here is basically just giving us an overview of what zinc can actually do for us, particularly in cardiovascular disease. And what you're seeing is that zinc has antioxidant effects by inhibiting different reactions with iron. So those are usually called Fenton reactions. It also protects against protein sulfohydryl groups. It's involved with, I didn't quote this, but metallothionein synthesis, glutathione synthesis, and then superoxide activity. And then it also has anti-inflammatory effects. So it will lower oxidative stress, lower inflammatory cytokines, and improve immune system functioning. All of this, by minimizing these different components, helps to minimize vascular and cardiac cellular dysfunction and thus minimize cardiovascular disease. So just having adequate zinc, or on the flip side, if you are zinc deficient or copper deficient or iron or vitamin C or vitamin E deficient, you can increase your oxidative stress levels. So again, any of the types of deficiencies can also drive that problem, and you wouldn't want to do that on the background of a high polyunsaturated fatty acid intake. Next, last one, I promise is the last one, is sleep deprivation. So sleep deprivation can induce oxidative stress. Basically, what they show here is collectively the data suggests that while short periods of sleep deprivation enhance antioxidant response in the brain and other organs, likely through a hormetic response, long-term sleep deprivation periods reduce the antioxidant response, suggesting that extended wakefulness induces chronic oxidative stress that lead to failure of antioxidant mechanisms to sustain the accumulation of pro-oxidants. Moreover, sleep dysfunction has been implicated in inflammation and immune dysfunction. In healthy individuals, acute or chronic sleep deprivation is associated with increased serum levels of interleukin-1, tumor necrosis factor alpha, interleukin-6, interleukin-17, and nuclear factor kappa beta, in addition to alteration of numbers and activity of macrophages and natural killer cells. In preclinical studies, sleep deprivation induced elevations in inflammatory markers such as IL-1, IL-6, tumor necrosis factor alpha, and peripheral blood and different brain regions also has been reported. So essentially, sleep deprivation or longer-term sleep deprivation can basically overwhelm the upregulation in antioxidant defense systems that you generally see in short-term sleep deprivation. And then that can drive oxidative stress. And the other thing is sleep deprivation also drives immune dysfunction And that immune dysfunction can lead to increases in oxidative stress, as we kind of talked about before, through like the nuclear factor kappa beta pathways, tumor necrosis factor alpha pathways, etc. And so basically what we're seeing here in this graphic is what I just described. So sleep deprivation is increasing oxidative stress. It's doing that through these immune markers and increases in inflammation. And then this has a negative impact on neurogenesis, learning, memory, and synaptic plasticity. And then there's also strong associations with shift work and sleep deprivation with cardiovascular disease and cancer. And it probably functioning through these oxidative stress pathways as well as these inflammatory mediator pathways, which can drive oxidation of cholesterol, oxidative stress at the endothelial cells, and then subsequent immune activation and formation of the plaques inside the arteries. So again, you have this other external factor with sleep deprivation on the basis of overloading your tissues with polyunsaturated fatty acids. Obviously wouldn't be ideal because you have highly peroxidizable fats in the face of an inflammatory oxidizing stimulus. So all together now, after going through all the different components, immune activation, exercise, stress hormones, exposure to toxins, metabolic dysfunction, nutrient deficiencies, and sleep deprivation can all increase reactive oxygen species stress. And now, if we have increased reactive oxygen species stress in the face of increased omega-6 polyunsaturated fatty acids and LDL, we have an increased production of oxidized LDL, which leads to damage to vasculature and increased immune activation and atherosclerosis. Now, something else to keep in mind is that increased reactive oxygen species stress can also drive damage to the vasculature and increased immune activation in and of itself, which can further drive atherosclerosis. And then some of the inflammatory mediators coming from the polyunsaturated fatty acids, they're called eicosanoids, can also drive damage to vasculature, increase immune activation, atherosclerosis. But usually these have a trigger from some of the immune activation and increased reactive oxygen species. So I give it a nice linear flow here, but there's obviously different connections to these different pathways overall. But this is the general idea that I wanted to get through. And then the last thing I wanted to show here is just, okay, what are we seeing in heart disease with the polyunsaturated fatty acids? So we discussed mechanisms and we discussed the interaction with the reactive oxygen species. So now, does this actually play out in heart disease? And so after we get through this, we'll talk about some solutions to this. So basically what we're seeing is that it has been known for decades that linoleic acid as a percentage of total fatty acids and lipids is reduced in patients with coronary artery disease. And this has been used as an argument to suggest that low intakes of linoleic acid may cause heart disease. So it's interesting, we're seeing, perhaps it's low intakes of linoleic acid, which wouldn't fit this theory. But then we go on to say, however, total fatty acid concentrations as opposed to percentages are independent of changes in other fatty acids and hence more reliable markers of linoleic acid intake, although likely less reliable compared with adipose tissue. And we'll talk about this in a future episode. Importantly, linoleic acid concentrations in both serum cholesterol esters and phospholipid fatty acids are in fact higher in patients with coronary artery disease compared to those without coronary artery disease. Again, since linoleic acid cannot be synthesized in the body, this suggests that patients who have heart disease consume more omega-6 linoleic acid than those without heart disease. So essentially, when you look at linoleic acid as a percentage, you see it perhaps decreases in patients with coronary artery disease. But when you look at it as its absolute concentration, which isn't dependent upon changes in the composition of other fatty acids, you're actually seeing that the linoleic acid composition inside serum cholesterol esters and phospholipids is actually higher in patients with coronary artery disease. So this actually fits the hypothesis that we're seeing overall. And basically what you're seeing is that people who tend to have coronary artery disease actually overall have that higher linoleic acid composition. So what they further say is, indeed, the authors of the study concluded cholesterol linoleic is widely believed to decrease in patients with coronary artery disease. Such decreases, however, represent decreases only in relative terms. We have shown in the study that linoleic actually is present in a higher concentration in individuals with cardiovascular or coronary artery disease than those without coronary artery disease. And basically this image here is just showing you guys what does this cholesterol linoleic actually look like. and you have this linoleic acid. So this is a fatty molecule of linoleic acid. And then you have cholesterol, and they're bound together. And since the linoleic acid is likely to oxidize and it will also oxidize cholesterol. And then you get a bunch of this will create the oxidized LDL cholesterol, and basically through these different mechanisms. And so I wanted to show the structure here. Next thing we see is oxidized LDL is higher in patients with heart disease. So oxidized LDL was found to be higher in patients with coronary artery disease compared with normal patients, and oxidized LDL was able to better identify patients at an elevated risk of heart disease. Moreover, oxidized LDL and autoantibodies to oxidized LDL are found in atherosclerotic regions. Furthermore, patients with progressive carotid atherosclerosis have more antibodies to oxidized LDL versus those without progression. So here we're seeing that oxidized LDL is directly involved in cardiovascular disease, and you're seeing that patients who have the progressive carotid atherosclerosis, and here I have a nice image of a carotid artery, and you can see the calcification, and you can see the narrowing of the artery in this particular region here, and that narrowing, that's showing the cardiovascular disease, that's showing the atherosclerosis. You can see the changes in lumen size, and they're showing the plaque formation and lipid core here using an MRI with, I think this is a gadolinium injection, so contrast. So pretty interesting overall. So we're seeing increased oxidized LDL, increased polyunsaturated fatty acid, including linoleic acid inside the cholesterol particles in coronary artery disease and heart disease. Next one, they say the most severe plaques in arteries have the most LDL. So what we're seeing is lipids from human atherosclerotic plaques have been found to contain oxidized cholesterol linoleic, so cholesterol esters containing linoleic acid. Moreover, the severity of atherosclerosis is noted to increase with increasing oxidized cholesterol linoleic. Healthier regions of aortas have been found to have less oxidized cholesterol linoleic, 5.8% to 9.5%, compared with atherosclerotic regions, which contain 12.4 to 21%. So essentially, what we're seeing is that oxidized cholesterol linoleic, which is the component that's going to oxidize this LDL cholesterol to a large extent, is higher in the aortas that have atherosclerotic regions. So you have the aorta, basically this is a picture of an aorta here with different atherosclerotic plaques on it. So the areas that have higher have higher amounts of atherosclerotic plaques have a much higher percentage of oxidized cholesterol linoleic in those particular regions. So giving us some extra data to look at some associations and see if these associations are supporting the hypothesis that we're putting forward. Now, the next one we talk about here is we're really focusing in now from just oxidized LDL and linoleic acid concentrations to oxidized linoleic acid. Specifically, because that was the main hypothesis. And what we're seeing is on LDL oxidation, linoleic acid is converted to hydroperoxides, which can then be converted to hydroxy acids such as 9-HODE, so 9-hydroxy 10,12 octodecadienoic acid. And then 9-HODE is extremely prevalent in oxidized LDL and is a good indicator of lipid peroxidation. In fact, 9-HODE is 20 times higher in young patients with atherosclerosis compared with healthy volunteers, and 30-fold to 100-fold greater in patients with atherosclerosis aged 69 to 94 compared with young, healthy individuals. So essentially, what we're seeing here is linoleic acid, when it gets oxidized, when it gets attacked by reactive oxygen species, it can form these different lipid peroxidation products, 9-HODE and 13-HODE. And essentially what we're seeing is that these components or these hydroxylipids or the linoleic acid components or metabolites attacked by reactive oxygen species are 20 times higher in young patients with atherosclerosis and compared with healthy volunteers, and 30 to 100-fold greater in patients with atherosclerosis in the 69 to 94-year-old group compared to young, healthy individuals. So we're seeing that oxidized linoleic acid metabolites are much higher in people with cardiovascular disease or atherosclerosis compared with healthy people who don't have that atherosclerosis, which is another association factor that supports the hypothesis. Now, last piece, or not the last piece here, I guess is the last quote here overall, but what we're seeing is we have increased oxidized linoleic acid metabolites means worse atherosclerosis. And so what we're seeing is in 1952, Glavland and colleagues published a paper showing that aortic lipid peroxides positively correlated with atherosclerosis. These findings were confirmed in 1970 by Brooks et al., who found large amounts of the 9-HODE and 13-HODE derived from linoleic acid hydroxyperoxides in aortic plaques. In 1991, Wang and Powell found increased amounts of 9-HODE and 13-HODE in the aortas and LDL of atherosclerotic rabbits. That same year, Belkner and colleagues found oxygenated cholesterol esters, specifically cholesterol linoleic, in atherosclerotic plaques of human aortas, the degree of which correlates with the stage of atherosclerosis. So essentially, more oxidized cholesterol linoleic in the atherosclerotic plaque, the worse the atherosclerosis was, or the later stage it was. To sum up, the increase in linoleic acid hydroperoxides in atherosclerotic plaques coincide with a greater severity of atherosclerosis versus normal regions. Basically, what I'm showing here is just what I was talking about initially when we were looking at the unsaturated fatty acids, was that the double bond areas are the areas most likely to be attacked. And so this is the linoleic acid being attacked by an oxidizing component, or oxygen specifically, or reactive oxygen species. And then this can form the hydroperoxide, either the 9-HODE or 13-HODE. I think this one is the 9-HODE, and it essentially just means that the hydroxide group is attached to the 9th carbon here on linoleic acid. And so this is just interesting to describe the process, because they're kind of describing these things here. But these are markers. The 9-HODE and 13-HODE are markers of the oxidized linoleic acid specifically. Now, what does this look like altogether? What is the overarching picture? Essentially, if we have a circumstance where we have immune activation, is that a viral infection, a bacterial infection, gut-derived endotoxin derived from periodontal infections, chronic UTIs, chronic bronchitis, etc.? Is there an increased exercise level? Is there a high amount of stress going on? Whether that's psychological stress, whether that's stress involved with upregulating cortisol or the catecholamines, or not having adequate calcium, or changes in blood pressure or sodium-potassium ratio? Is there exposure to toxins? Is that heavy metals? Is that particulate matter in the air? Is there metabolic dysfunction? Is that diabetes? Is that obesity? Are there nutrient deficiencies? Is that B vitamins, vitamin C, vitamin E, zinc? Are you sleep deprived? In any of these circumstances, you're likely going to experience an increase in reactive oxygen species stress. So you have the fuse here. These are all the fuse. These are all situations that a lot of us are going to be exposed to over the course of our life. They're relatively ubiquitous for most people. And now the body has to handle this reactive oxygen species stress. But if the cells, the cholesterol components and whatnot are loaded in polyunsaturated fatty acids, well, now you have fatty acids that are likely to peroxidize. In this case, we specifically talked about omega-6 polyunsaturated fatty acids in the form of linoleic acid. So what you can think of is these components, the reactive oxygen species stress is the spark. They're lit by these different lifestyle components that we would experience. And then the amount of polyunsaturated fatty acids that we load into our tissues, our cholesterol components, etc., is essentially the dynamite that adjusts our susceptibility to the reactive oxygen species stress, the stress that we experience for multiple components of our everyday life. So the solution to this overall picture would be to minimize your reactive oxygen species stress by adjusting these various factors, and then also to not load your tissues with the polyunsaturated fatty acids. And so that gets us to our favorite part here, the solutions page. Now, a lot of these components, some of these dietary components, I actually do cover directly how to set up your diet and directly how to organize the polyunsaturated fat intake, the ratios between omega-6 and to omega-3, how to hit your various micronutrients, your vitamins and your minerals, how to manage your stress effectively. I cover all of these things in a bit more depth inside the nutrition blueprint, which is essentially a guide that I created. It's on my website. I will link that guide into the description below, but just as far as specific solutions for everyone. So what's the best way to minimize the amount of polyunsaturated fatty acid you have in your tissues so that you're less susceptible to reactive oxygen species stress? Well, decrease your polyunsaturated fatty acid intake. Now, I'm not going to go into the specific studies in the interest of time because this video is already quite long, but generally the goal would be around 4 grams per 2,000 calories per day. If you're a little bit over, no big deal. If you're under, great. But this is just a ballpark estimate. The next thing we want to do is we want to decrease omega-6 intake in ratio specifically with omega-3 intake while still keeping total polyunsaturated fatty acid intake low. So now this doesn't mean that supplementing fatty like fish oil or eating tons of fatty fish I think is a good idea. And in future episodes, I'm going to cover problems with omega-3 fatty acids. And then Jay and I will also be releasing a podcast on the on our energy balance podcast discussing problems with omega-3s. But essentially, you want to shoot for like maybe around a 3-to-1 ratio. So maybe you would have 1 gram of omega-3 in a day and then 3 grams of omega-6. And there's some adjustments in inflammatory signaling here. And essentially, this is based on how I set up the diet and the nutrition blueprint. You'll already see how to actually do this. So by all means, go check that out. It's very specific on how to set these things up. Then the next thing is you want to make sure that you have adequate antioxidant buffering capacity to deal with that ROS, kind of like how we discussed when we were discussing polyunsaturated fatty acids earlier, where adequate antioxidant systems are super helpful for mopping up the excess ROS so that the fatty acids and other components of your cells aren't exposed to them. So that includes having adequate B vitamins or adequate vitamins in general, which includes the B vitamins, vitamin C, vitamin E, vitamin A, vitamin D, vitamin K. They're all involved in multiple processes that help with antioxidant function. That also involves having adequate minerals, copper, zinc, iron, selenium, manganese, magnesium, calcium, etc. Now, I know they said copper and iron can increase oxidative stress in some of the studies we discussed, and they can, but they also are directly involved in different enzymes that are important for minimizing oxidative stress. You want to make sure you have enough of those, just not too much. Then we also want to include protective plant compounds. There's many polyphenolic compounds that have pretty potent antioxidant effects, so we want to incorporate those. Now, the other thing we want to do is we want to minimize our exposure to different things that are going to increase ROS. And some of those we discussed throughout, but that's smoking, pollutants. So pollutants include BPA, BPS, phthalates, the particulate matter we discussed, heavy metals, so mercury, lead, cadmium, arsenic, etc. Excessive exercise, so way too much exercise. Then we want certain drugs can increase ROS pretty significantly. Some of the antibiotics, for example, like fluoroquinolones and Flagyl or metronidazole, both can increase ROS and cause issues in the mitochondria. And then we have radiation. There's many other components, but those are some basic ones to consider. And what I want to mention here is that there is a limit to the body's ability to mop up oxidative stress. There's only so much that the antioxidant enzymes systems can handle. And this is part of the argument that Jay and I have talked about in terms of hormesis. So I would recommend checking out some of the hormesis series in the Energy Balance podcast. And I will be doing future episodes discussing hormesis and some of the systems around there, and then managing stress and allostatic load and things from that perspective. But it's not like you can just continually increase these things that induce protective pathways of the body and think that the body has unlimited capabilities to handle that. So it's something important to consider, that you can overload these systems. For example, you can excessively exercise or excessively work yourself to death, essentially, even if you have adequate nutrition and all these other components on board. So there is a need to limit that. Or even if your nutrition and your supplementation, your exercise and sleep are great, if you're exposed to certain pollutants or heavy metals or certain toxins or drugs, they can still have a very significant negative impact despite all those other factors in place. So that's something to consider, that you really want to make sure that you're avoiding things that overload the system beyond its capacity to recover. Now, the next piece is to make sure that you're managing stress effectively. So there's multiple things that I mean by stress, and just some 3 basic examples is having adequate fuel on board. So that's carbohydrates and fats. And essentially, this is looking at stress through the lens of not having enough energy to meet a demand. So you want to make sure you have enough fuel, and that the actual energy, the production of energy from that fuel is appropriate, so that you can produce enough energy to meet a demand. So you want to make sure all of that is functioning well. The other thing is you want to make sure that you have effective philosophical and psychological coping strategies to minimize your exposure to stress, or to the production of these stress hormones, adrenaline, cortisol, glucagon, etc. And essentially perception of different events, life events, whatnot, and how you manage or deal with those things can affect how much of these different hormones you produce. So it's important to have both your actual empirical dietary and supplementary things set up appropriately, so that you have the adequate energy. And then it's also important to have the correct mental frameworks and philosophies on dealing with different circumstances, so that you can divert and adjust that energy in an effective manner, both consciously and subconsciously. So that's kind of number 1 from the stress perspective. The next level of stress is calcium stress, or not having adequate calcium or too much phosphorus. So essentially what we want to do is we want to make sure there's enough calcium in ratio to phosphorus. We have enough magnesium, enough vitamin K, enough vitamin A, enough vitamin D to minimize the effects of excess parathyroid hormone, which directly is involved in cardiovascular disease and the production of ROS of the vasculature and atherosclerosis. And so we want to make sure we're keeping parathyroid hormone in safe levels or effective levels. Usually I'm looking at blood tests to be less than, I think it's 30, I think the units are picograms per milliliter, if I remember correctly. And then the next one is minimizing that angiotensin II cascade, or that RAS cascade. So we want to make sure we have adequate sodium, potassium, and fluid intake on board to minimize the excess activation of the RAS system, which can drive the antidiuretic hormone, angiotensin, endothelin-1. We didn't talk about antidiuretic hormone and endothelin-1 inside vascular dysfunction and whatnot, just for the sake of time, because there's like endless number of mediators to talk about. But we want to make sure that we're not putting the system under that type of stress and rapidly increase or drastically increasing those process and those signaling, because the angiotensin II and the endothelin-1 and all these different components are pretty damaging to the vasculature in and of themselves. The next thing we want to do is we want to make sure that we're getting adequate sleep every night. So that's 7 to 9 hours of sleep per night, and we want to make sure that it's actually quality sleep. And we want to make sure that it's consistent. So that's both dietary, supplementary, and lifestyle strategies to optimize your sleep. There's very specific components there. I will be creating a guide in the future over just some of the basics. So you can, when I release that, you'll be able to find them on my website. Then the next thing is managing the allostatic load, or the stress load of your exercise. And so an important piece to consider here is there isn't a minimum effective dose of exercise, as well as an excessive level of exercise where the body can't recover from it. And you're going to want to find out which level is appropriate for you, your goals, and your context. And the other thing is, with the context component, there's individuality to this. So some people can handle exercise quite well. Other people, it takes a bit more of a toll on them. And then some people are working 60, 80 hours a week. So they're not going to, it might be quite difficult to get to run a marathon, which I wouldn't recommend anyway, but to like go to the gym and do some type of structured exercise 6 days a week. So you really want to be adjusting your exercise load to your general stress load, and going from there so you're not overloading the system. The next piece is addressing anything addressing any dysbiosis or microbial disturbances inside the intestine, which can lead to intestinal permeability and increased production of gut-derived endotoxin and other microbial metabolites, which directly increase ROS, directly increase immune and inflammatory signaling, and are strongly associated with increased risks of cardiovascular disease. Again, I'm not going to go through all those studies in this PowerPoint. I am looking to cover endotoxin and gut-derived components on cardiovascular disease in another presentation. But essentially, we want to make sure the gut is functioning well and the microbiome is situated appropriately. Another one is minimizing chronic latent infections. So there are some associations between cardiovascular disease and different latent infections. Some of those can include having infections in your teeth. So periodontitis, you want to make sure that there's not a lot of issues going on with oral health, because it's essentially the damage to the gum structure and bacterial integration or invasion into those areas can lead to leakage of endotoxins chronically low level that can stimulate that oxidative stress and stimulate the inflammatory cascades at the vasculature. Chronic UTIs, same deal in the bladder. Chronic bronchitis, same issue in the lungs. Or chronic sexually transmitted infections can also lead to increases in risk of different types of disease. So make sure that, if there any of those things are going on, that they are directly addressed. Then the last one is a very overarching one, but making sure to correct underlying metabolic issues. We want to make sure that there's not an excess reliance on fatty acid oxidation and an inability to adequately oxidize carbohydrates effectively. This runs in the face of the low carb dietary philosophies, and it's their ideas of metabolic flexibility, as I kind of covered previously, are actually counter to what's actually discussed in the research, particularly in states like diabetes, which are characterized by an inability to adequately oxidize carbohydrates with an upregulation of fatty acid oxidation. So we don't want to be driving that. Fatty acid oxidation in and of itself increases ROS production at the mitochondria. I discussed the mechanisms in my Randle Cycle video. So we want to make sure we're having adequate carbs and that we're effectively handling any type of metabolic dysfunction, and whether that's a pre-diabetic, a diabetic state, an obese state, impaired glucose tolerance states, etc. And now something to consider is many of the steps that I discussed above will also help to manage these problems with metabolic issues overall. And again, there's more to those pictures, but trying to address those effectively will go a long way into minimizing oxidative stress. So hopefully this video was helpful for you guys. I laid out a lot of different mechanisms. I talked about different theoretical components. We supported it with some associative research on the back end, and then now we have some solutions here. So if you like this content and you want to see more, of the cardiovascular series, give me a like, give me a subscribe, and perhaps comment and let me know what you want to see below. I'm looking forward to producing more content on a consistent basis now that I'm a bit more settled. Look forward to seeing you guys in the next one. Hope you enjoy the presentation.